Welcome back to my channel Biochem Bio World. Today is a video of the extension of the last video, and basically I'll be discussing the functions of the structural components of the retrovirus, that is HIV, human immunodeficiency virus. Okay, so let us begin with it. And this is a table, a summarizing form of the structure and proteins which are structure components of that particular virus, the genes which are involved in the synthesis of these structure proteins, and what are is the, we can say, the main function of that particular structure protein in a virus. Okay, let us begin with the surface proteins, and usually there are two surface proteins, and gp160, gp120. Gp here is meaning the glycoprotein. Okay, the env gene is responsible for the synthesis of both the surface proteins, and the functions are different from them. gp160 is a precursor of the envelope protein, that is the surface proteins and the transmembrane proteins, whereas gp120 is responsible for the attachment of the virus to the target cell, or we can say the host cell. This is the main we can say the surface protein which is responsible for the infection, that is for the infection to the host cell. gp120 is necessary. And next comes the transmembrane proteins, that is gp41. It is synthesized by the env gene, synthesized from the env gene, and it is responsible, that is it is an anchorage of the gp120, and it usually results in the fusion of viral and cell membrane. It is responsible for the fusion of viral and cell membrane. Okay, next are the enzymes. I talked about three enzymes are present, that is reverse transcriptase, RNase H integrase. This is basically a complex, and we know that the HIV virus is having RNA as a genome, and the target cell is having DNA. So it is not directly like the RNA cannot get integrated into the DNA host genome. Okay, they are not compatible if you think. So the viral RNA is usually transformed. It is usually, we can say, transformed into the DNA, and which is usually termed as a proviral DNA, which gets integrated into the host genome. Okay, and the enzymes, the three enzymes which is responsible for whole this process is reverse transcriptase, RNase H, and integrase. Reverse transcriptase protein 51, and these three basically are enzymes. Uh, they are synthesized from the Pol gene. Okay, and the function of the reverse transcriptase is it is synthesized of the proviral DNA from the HIV RNA. Okay, and during this process, the synthesis of the proviral DNA, what happens, there is an RNA DNA intermediate, okay, which is it is not stable, and the viral RNA is usually degraded, and so as to synthesize the double stranded proviral DNA. Okay, the degradation of the viral RNA is usually carried by the enzyme RNase H. Okay, and the integration of that proviral DNA which is synthesized at the end of this phenomena is carried out by the integrase enzyme, that is P32 protein 32, and it helps in the integration of that proviral DNA into the host region of the target cell. Okay, next is the capsid protein P24. It is usually synthesized by the GAG gene, and it is usually responsible for the formation of the conical capsid which is surrounding the genome. Okay, next is the matrix protein. There are proteins which are also present in the matrix. Matrix means the cytoplasm of the virus, and is usually protein 17, and the GAG gene is responsible for the synthesis of this protein, and basically it is a myristylated protein. By myristylation, that is, it is lipid, that is myristic acid is attached to that protein and is usually forming the inner membrane layer. Okay, next is a protease. A protease enzyme is also present there, and as the name suggests, it may be carrying a proteolytic cleavage, and is usually helps in the release of the structure proteins and viral enzymes. Okay, it does release structure proteins and the viral enzymes. Okay, and next is nucleic acid binding protein and the link protein. These are two other proteins, and they are usually synthesized by the GAG gene, and the nucleic acid binding protein is usually, it is result in formation of nucleoprotein and RNA complex. Okay, there may be some proteins which are they are in our genome, in our DNA. Usually it is bound by histone proteins, so the same goes for here. And the protein RNA complex, it is carried out P7, that is nucleic acid binding protein, and which is synthesized by the GAG gene. Next is the link protein, and P6, it is synthesized by the same gene, GAG, and it is responsible for the release of the virus particle from the cell via lysis. That was all about the functions of the structure proteins. Thank you so much for watching my channel. Please like, subscribe and share.